Moving on, we have our last presenter. We have Judy Xia.
Morning, everyone. My name is Judy, and today I'll be presenting my research topic on optimizing culture media and digestion protocol for the tumor macrophage co-culture system on tracer. So for this presentation, I'll start with talking about the motivation behind our research, which is to mitigate the poor prognosis results of the pancreatic cancer. Then I'll introduce you to a 3D tumor model called tracer that we use to conduct our investigation. Throughout this workflow, we encounter two distinct problems, so consequently, I'll talk about the methodologies we use to solve the problems, as well as the results we obtained. At the end of the presentation, I'll show you the impacts of this research and tie it back to the bigger picture. So pancreatic ductal adenocarcinoma, also known as PDAC, is one of the most lethal cancers in the world, with a 5-year survival rate of only 7 to 8%. The poor prognosis is mainly correlated to the lack of effective treatments due to the tumor microenvironment. So how does the microenvironment contribute to the poor prognosis of the patients? Well, the answer lies within the high heterogeneity of this microenvironment. In this highly dense tumor mass, we have a wide variety of cell types that are constantly interacting with the cancer cells, aiding the cancer progression. Among these cell types, we want to focus on the interactions between the macrophages, which is a type of immune cells, and the PDAC tumor cells. Now, the reason we chose to look at macrophages in specific is because that when in this microenvironment, they're said to be activated and assumes a pro-tumoral state. So they play a substantial role in, for instance, angiogenesis, immune suppression, tumor proliferation, and metastasis. And this is exactly the reason that we want to construct a co-culture system between the PDAC tumor cells and the macrophages to identify the interactions that we can use to reverse the effects of the macrophages. Now, the McGuigan lab has developed a 3D tumor model called tracer that has successfully established the nutrient and oxygen gradient while also recapitulating the in vivo tumor microenvironment. The setup of tracer is actually quite straightforward. We start with seeding the cells on tracer scaffold. Then we roll it along an oxygen impermeable core. After culturing, we unroll the tracer and isolate cells for analysis. So the compartmentalization of tracer provides us an ideal platform to study the co-culture system. Now let's take a look at the timeline of this workflow. If we take the day of rolling tracer as day 0, then 2 weeks before that, we start culturing the primary PDAC cells. On day -5, we see the tumor cells on the tracer scaffold for 4 days using the PDAC growth media. On day -1, we add the macrophages to establish the co-culture system. The rolled tracer will be cultured for 3 days, and on day 3, the cells that are trapped within the scaffold will be dissociated out using enzymatic digestion and will perform downstream analysis. However, there are 2 different problems that we encounter in this workflow. The first problem is that the co-culture media we use on day -1 is 100% PDAC growth media, and this media itself activated the macrophages even before the tumor cells comes into play. Now, the second problem we have is that on day 3, the digestion protocol we use resulted in low yield and low viability of the cells. Both of these problems can bias our downstream analysis, and without appropriate solutions, we'll have a bit of problems. Now I'll talk about the methodologies and the results we obtained for co-culture media optimization. So the idea is that if it's a PDAC growth media that gave us a problem, why not switch the co-culture media to the 100% macrophage growth media? And that will allow us to bypass the activation problem. But we also need to ensure that the tumor cells can survive the sudden media switch. So what we need is a buffer zone between day -5 and day -1 to prepare them to adapt to the media switch. We decided to create this buffer zone by turning the PDAC monoculture media into a combination of the PDAC growth media and the macrophage growth media at different mixing ratio. 5 different media combinations were tested, and for instance, 25 RPMI stands for 25% of the macrophage media and 75% of the PDAC growth media. And the experimental procedure is as follows. On day 0, we see the tumor cells on the tracer scaffold using one of the 5 testing conditions. On day 4, the media are switched to 100% RPMI, and the growth is continued for another 2 days. The tumor cells is stained with CK19, which is a maturation marker, as well as DRAC5, which is a nuclear stain. Fluorescent confocal images will be taken on day 4, 5, and 6. And our hypothesis is that the optimal monoculture media will lead to an uphill trend of SECA19 expression on day 5 and day 6. Now, the mean gray value of SECA19 from these fluorescent images can serve as a perfect indication of the PDAC cell's maturation stage. And on the right side, we show you the results of the 5 different testing conditions. Here we see that for 75 RPMI and 25 RPMI, an upward growth trend can be observed. And therefore, we conclude that these mixing ratios are the optimal monoculture media. And for future results, we will test more replicates to ensure the reproducibility. Next, we'll move on to phase 2 of the project, where we will optimize our digestion protocol. Now, recall that on day 3, the enzymatic digestion are required to isolate the cells from the tracer scaffold for downstream analysis. But the problem we have is that the current protocol leads to a low yield and low viability of the macrophages. After reviewing the literatures, we determined on the 4 parameters to optimize. For enzyme types, we selected collagenase 11, collagenase 4, and liberase as they were commonly used for tissue dissection. And for enzyme concentrations, we also determined 2 optimal ranges for collagenase and liberase. We also decided to investigate whether incubation time and mechanical shaking played a role in our digestion results. Our hypothesis is that a strongly digestive enzyme with lower concentration and shorter incubation time will give us the most optimal results. So here goes the experimental procedures. We see the macrophages on tracer scaffold for 2 days. Then we run the enzymatic digestion. Now, the digested fractions will be analyzed by a plate radar, and the fluorescence emission of the calcium and propionate iodide can help us to calculate the live to dead cell ratio. A total of 8 trials were investigated to test the 4 parameters that we want to optimize. So here on this graph, we show you the relative proportion of the live to dead cells of the 8 trials. You can see that all the trials of the liberase and collagenase 11 resulted in high live cell counts. Trial 4 is slightly better than trial 5, presumably due to the lower concentration. And all the collagenase 11 trials performed surprisingly better than our previous testing results, and we believe it can be attributed to the shorter incubation time and the absence of mechanical shaking. On the contrary, collagenase 4 plus liberase combination resulted in high dead cell counts, and we believe that this combination may be too harsh for the macrophages. So in conclusion, we investigated in 2 problems that we encountered when establishing the co-culture system on tracer. For the lack of media optimization, we concluded 2 different mixing ratios that are the optimal monoculture media. And in future steps, we will run more replicates to test the reproducibility of the results. For the lack of digestion optimization, we determined that the liberase and collagenase 11 are the 2 more ideal enzyme types. We also determined the optimal concentration range, incubation time, and the shaking condition. And for future, we'll further optimize the protocols based on the current findings. Now, I'm pretty sure you have heard of me talking about downstream analysis many times in this presentation. Now, what does it really entail? For the cells that are digested out, we will run single-cell RNA sequencing on them. And this is a technique that has an enormous field of application. For example, we can use data extracted to identify differentially expressed genes, upregulated gene pathways, as well as clonal diversity. So it really is the enabling of all these downstream analysis of this current research project that paves the path to the vast potential to the overall project. With different pieces of valuable information coming together, we hope eventually that we can identify the interactions between the PDAC tumor cells and the macrophages and use those interactions as a therapeutic target to treat pancreatic cancer. And with that, I'd like to close off by thanking Professor McGuigan and Ileana for supervising my summer research project, as well as Department of Chemical Engineering and Applied Chemistry for funding my research through the UTA scholarship. Thank you for your time, and I'd like to take on any questions that you might have. Thank you.
Thank you, Judy, for your presentation. We now ask if judges have any questions, please to unmute themselves.
Sorry, thank you very much for your presentation. I was wondering if you could go to page 13.
Yep, for sure.
30, sorry, 30.
Oh, 30, okay. Yeah.
So you mentioned like these enzymes were like, well, like according to your optimization, like digestion optimization, they were like good. And I was wondering if you could elaborate on the reason, like why you chose like these two enzymes.
Yeah, absolutely. So there are essentially two criteria that we want to look at, right? So one is the viability of the cells, and one is the yield. So perhaps how many cells are digested out of the scaffold. So the number one criteria we use here is the viability. And here you can see, so here is the fluorescence stain that we did. And from the fluorescence emission, we can calculate the relative proportion between the live cells and the dead cells. And you can see that the librases and collagenase both gave a higher live cell count comparing to the dead cell, whereas collagenase 4, so this is the enzyme type that we decided not to go with, had an extremely high dead cell count. So once we decided that so on slide 30, when decided that these two are the more optimal enzyme types, we actually ran a third round of the experimentation using different concentration of the librases and collagenase 11. So here I can show you the results. So as you can see, once we optimize these two enzyme types, so we run another 11 trials using, so here are the conditions you can see. For example, we use the libraries, and for example, collagenase 11, we adjust the incubation time, we adjust the shaking condition, and these are the new results. And as you can see, the viability of all of them is almost 100%. So none of them are very harsh to the cells. And now we look at the yield, right? So you can see that trial 3 to 5 and trial 10 has a better yield. So that means that they can digest more cells out of the scaffold, so which is good because we want to run the RNA sequencing analysis. So we decided that the collagenase, either with or without libraries, but collagenase as the primary digestive enzyme is the best choice for high yield of the macrophages.
Okay, thank you very much. So basically in here, you did like decoupling between like the factors, the processing conditions, right?
Yeah, so essentially we compare one factor at a time, right? So for example, here, so we compare the different concentrations, and so someplace, for example, here we have a baseline condition, right? So for example, we can compare this one with trial 7. So all the conditions are exactly the same except the time and the shaking speed. So for shaking speed, we have a different set of experimentation to explore that. So that's in our round 2. So you can see here. So we run different rounds of optimization experiments, right? So these ones are for us to test the concentration. So that's the only variable we changed. These ones are for us to test the time. So that's also the only variable we changed, and types, as well as the mechanical shaking here. So some of them have shaking speed and some of them don't.
That is great. Thank you very much.
Yeah, thank you for your time.
Thank you, Judy, and thank you to our judges for those questions.
Thank you.
Okay, thank you everyone for your time. This concludes our podium session. Judges, if you have filled out the rubric through the online judging form, our judging director will be compiling the marks for these presenters soon. Thank you so much, everyone, for joining us.